Good evening, dear students. In this lecture, I will talk about the fossil members of Pteridophyta. There are 5 fossil members that have been included in your syllabus: the Rhynia, Zosterophyllum, Psilophyton, Lepidodendron, and Calamites. The first 3 belongs to the Psilopsida group, and the Lepidodendron belongs to Lycopsids, and Calamites belongs to Sphenopsids. So what are these Psilopsids, Lycopsids, or Sphenopsids? Let's first understand that with the help of a classification given by K.R. Sporne in 1975. I have chosen this classification because it is an easy classification. It gives a gross idea about into how many groups the pteridophyte can be divided, and that's why I have included this in this lecture. Though this is not there in the syllabus, but K.R. Sporne's classification is basic one. So let's begin with the classification first, then we will enter into the details of the genera. Okay? So K.R. Sporne, 1975, he classified pteridophytes into 6 classes. Class 1 is called Psilopsida. Psilopsida, it includes 3 orders: Trimerophytalis, Rhynialis, Zosterophyllum, and all these 3 are extinct. None of the members of these is living. These are living. From each of these 3, we will study one genus. We will see that. Class 2 is Psilotopsida, which is also called whist fern. And this is a living member. Some are extinct, some are living. Psilotelis is the order within this class, and the Psilotum is the living member of that. Timacipteris is another living member of that. The Psilotum is possibly there in the syllabus as far as the living members are concerned, and that part will be taught possibly by AM sir. Class 3, it is Lycopsida, the most popular one that you know. The Lycopodium and Selaginella belongs to this particular class. There are following orders: Proto-Lepidodendron, which is absolutely fossil. Lepidodendron is also fossil. Lycopodialis is extant, living, as well as some are fossil also. Isoetelis, which includes the Isoetis, which is living. Selaginellalis, which includes Selaginella, is living. Class 4 is Sphenopsida, also called horsetails, and there are 4 orders within this: Rhynialis, which is fossil; Sphenophyllalis, fossil; Calamites, fossil; and Equisetalis. Among these orders, we will study Calamites from Calamites, and Equisitum, which is a living member, from Equisetalis. All these are basically Sphenopsida groups. Anyway, Pteropsida is the fern group, and it includes a big group. Most of the pteridophytes belong to this Pteropsida, or Phelicopsida also called, and there are 4 orders. These are subclasses. In fact, there are many other orders. The subclasses are Primophilices, which is absolutely fossil. It includes Cladoxylalis and Synopteridalis. And the subclass Eusporangiati, it includes Meratialis and Ophioglossalis. These are living. Osmandalis is also living. The subclass is Osmandidi. And Leptosporangiati, it includes the Felicalis, Marcialis, and Salvinialis. This Felicalis is the true fern group. The Dryopteris, Pteris, the Cyathus, etc. belong to this. Those particular genera, they belong to this Felicalis order. And the 6th class, Progymnospermopsida. This K.R. Sporne has kept under pteridophyte as a 6th class, but most of the scientists, they treat Progymnospermopsida as a separate division. Just like Pteridophyta, it's a division in between Pteridophyta and Gymnosperm. It is called Progymnospermopsida, and this particular group is the transition between the pteridophytes and the seed plants or gymnosperm. And none of the progymnospermopsida are living today, all are extinct. And they are very interesting plant. People may ask the missing link between the Pteridophyta and the seed plant. So these are the missing links, progymnospermopsida group. The most important genera that is included in your syllabus is Archaeopteris, from this order, Archaeopteridalis of progymnospermopsida in your syllabus. So now moving to the So this is the overall idea about the different classes of gymnosperm. You should remember this like Psilopsida, Psilotopsida, Lycopsida, Sphenopsida, and Pteropsida. 5 classes. You should remember that Pteridophyte can be divided into 5 groups. In general, if you say groups, there is no problem. So there are 5 groups. Like you remember algae to be classified into 11 classes, of which the main classes are like Chlorophyceae, Xanthophyceae, Phaeophyceae, Rhodophyceae, Bacillariophyceae. You remember like that, na? You also remember the fungi classified into 4 classes, like the Phycomycetes, the Ascomycetes, Basidiomycetes, and Deuteromycetes. Like that, grossly, you should have an idea that how many groups. But there are other classifications also, which are further divided these into different orders and classes. So this is a general classification to keep in mind. So now we will concentrate on Psilopsida class, within which 3 genera, those have been shown in yellow. The Rhynia, Psilophyton, and Zosterophyllum has been included in the syllabus, and they belong to the Rhynia belongs to the Rhynialis order, family Rhyniaceae. Rhynialis order, in fact, there are 2 families, Rhyniaceae and Cooksoniaceae. Cooksoniaceae, it contains the Cooksonia genus, which is considered as the first land plant, first vascular plant, known vascular plant, first known vascular plant. You can say there may be, we might not be knowing some plant which evolved before that, which originated before that and had become extinct. We might not be knowing, but what we know by the fossil record is this is the first land plant, Cooksonia caledonica. Rhynia, Horneophyton, Yaravia, they are the members of Rhyniaceae family. All these plants are, in fact, the entire Psilopsida class, they are the first land plants. When did this phenomena took place? When the first land plant, the plant before that were aquatic in nature, semi-aquatic. The bryophytes, from bryophytes, pteridophytes have come. If you believe in progressive theory of evolution, there are retrogressive theory of evolution also. Some scientists say that no, pteridophyte have come first, then bryophytes have come. So this is retrogressive theory. And progressive theory, most people believe in that progressive theory, that okay, the bryophytes have come from the algae, and from bryophytes, pteridophytes have come from pteridophytes, progymnospermopsida, and from progymnosperm, gymnosperm, and then angiosperm. So this is the line of evolution. And it is, we can also correlate with the structural development or complexity that we find in these groups. So this entire class Psilopsida are the first group of land plants that came on the earth nearly about during the period Silurian, middle Silurian to Devonian, that period, about 40 crore to 35 crore years ago, if you talk in crores of years. Okay. Now, So you may note this, that the Cooksonia is the first land plant, and Rhynia, Horneophyton, all these, the Trimerophyton, Psilophyton, Pertica, Zosterophyllum, 3 have been included in your syllabus because everything cannot be there in the syllabus. It will be too big. In masters, you will have to read, study all of them. So all these plants are the first group of land plants that evolved from the aquatic habit. And the time period is Silurian to Devonian, middle Silurian, in fact, to Devonian. The Cooksonia, it was there during middle Silurian, and it became extinct during early Devonian. Rhynia, Horneophyton, Horneophyton is little earlier than Rhynia, and Rhynia was there during middle Devonian. Psilophyton and Zosterophyllum are little bit before Rhynia, in fact. So that was the time period when plants were evolving from aquatic to land habit. And all these plants are rootless, leafless. Remember, rootless, leafless, extinct member of Pteridophyte. Sometimes questions are asked like that. Describe the sporophyte, the morphoanatomical features of a rootless, leafless plant belonging to the order Rhynialis or Rhyniaceae, or included in your syllabus like that. It may be like that. So in that case, you have to understand which plant they are talking about. Trimerophytalis, the name is such because they had a trichotomous branching pattern. This Trimerophyton and Pertica, it showed trichotomous. These plants had dichotomous branching pattern, and this Trimerophytalis had trichotomous branching pattern to some extent. Psilophyton was, however, dichotomous with a little bit of, you know what is dichotomous? The branch goes and it divides into 2 equal halves. That is dichotomous. If one of the half overtops the other, it becomes monopodial branching pattern, and that is a little bit of that is seen in case of Psilophyton. We will see that. Zosterophyllum also shows a kind of dichotomy. So now let's see one by one. Rhynia, I already have taught the major, the honor students. That will be repeated a little bit. You have to stay calm during this period. Rhyniopsida, first the characteristic of the class. Here, Rhyniopsida, it has been treated as a class. This is not a, mistake. There are other scientists which designate Rhynialis. They lift it to a class level, Rhyniopsida. So we can treat it as a class, or we can also write Rhynialis here. The general features are plants bore terminal sporangia on their aerial axis. Sporangia usually are ellipsoidal, but they may be branched or reniform, as in Cooksonia. In Rhynia, you will see ellipsoidal sporangia, but in Cooksonia, you will find reniform sporangia, and these are terminal in position. Dehiscence, longitudinal. However, this is about Rhynia. In Cooksonia, the dehiscence is somewhat, you can say it is transverse. Branching, dichotomous. Aerial axis, terete, means circular in outline. Stele, protostelic. Generally haplostelic. So it is one of the most primitive stellar type, the protostele, and among that, haplostele is the most primitive one. Now, talking about Rhynia, the plant Rhynia, how does it look like? It looks like this. So here, this first plant, this one, this is Rhynia major, and this is Rhynia Goinivoni, Gwynevaugani, Goinivoni, you can say, Rhynia Goinivoni. This one. This was about 18 to 20 centimeter tall plant, very small, 18 to 20 centimeter. You can imagine little bit about 7 inches, 7 to 8 inches tall. And the Rhynia major, which is no longer Rhynia, because we will see that Edwards in 1986, he has shifted this Rhynia major from this genus to another genus called Lepidodendron major. So Rhynia major, it was described as a Rhynia when this plant was discovered first. Rhynia major was first described. It is a tall plant, more or less about 50 centimeter or so, about 60 centimeter or so. The plant has the Rhynia major as well as the Rhynia guinevere. So we should talk about Rhynia guinevere because Rhynia major is no longer Rhynia, it is Lepidodendron major now. So we will concentrate on this plant, this middle one. And this plant, this is Horneophyton, so you should not concentrate on this particular diagram, this diagram, this diagram, this diagram. You omit these 3-4 diagrams, okay? And we can concentrate on this. This is Cooksonia. Cooksonia looked like this, dichotomously branched. The lower part was not found because this is fossil. Only the upper part has been found as a fossil record. And there are terminal sporangia, which are reniform, kidney-shaped. You can see this is the structure of the sporangia of Cooksonia. Cooksonia is the first land plant, remember that. And this is the suture through which dehiscence took place, and it is transverse suture. And this is sporangia. This is the sporangia of Rhynia major, as well as first described as Rhynia major sporangia. But Rhynia major and Rhynia guinevere, they have similar structure as far as the sporangial outline is concerned. Overall structure is concerned, they're similar. Size-wise, they're different. So Rhynia guinevere, there is a horizontal rhizome from which the aerial axis arise, and that goes upright and it divides dichotomously later on. Some of the branches bore the sporangia, some branches did not bear any sporangia. And there are some other branches coming from the sides. These are called adventitious branches. And these branches possibly used to detach from the plant, and just like you can say that a bulbil or a bud, they can give rise to a new plant. So it kind of a vegetative propagation these particular adventitious branches used to show. So this is the structure of Rhynia guinevere plant. It is a dichotomously branched plant. This particular dichotomy, the terminal dichotomy, is called telome. You possibly have heard about the telome theory. So telome theory talks about the terminal branch of the dichotomy of Rhynia, from which all other different types of plants, the structure of plants evolved. The microphyll, the megaphyll, the stem, the seed, everything, the seed, the integument of this seed, everything originated from this telome type of structure. There is a theory regarding that given by Zimmerman. So let me go to this one. So when was this plant discovered? 1913, discovered by Mackey, but described by Kidston and Lang in 1917. So Kidston and Lang, they are given credit for describing Rhynia major as well as Rhynia guinevere. And it was found in the Rhynie chert beds of Scotland. Geological age was late Lower Devonian to Middle Devonian, approximately Middle Devonian. Initially, two species named and described, Rhynia major and Rhynia guinevere, but later D.S. Edwards changed the name Rhynia major to Lepidodendron major. You should remember also this. You might be asked this short question: what is the new name of Rhynia major? Lepidodendron major. Who gave this name? D.S. Edwards. But Lepidodendron major is not a vascular plant. It is non-vascular plant. It was wrongly described by Kidston and Lang as Rhynia major. when it was analyzed further, studied further in details, it was found that it should not be kept in. It is not in fact a vascular plant, it is a non-vascular plant, and its name was changed to Aglaophyton major in 1986. So now only one species, Rhynia gonivoni, is considered under this genus. So Rhynia gonivoni is characterized as follows: morphological characteristics. I already have said many characteristics, but let us have a glance. The plant body was sporophytic. This is a sporophyte of the plant. We will talk about what was the nature of the gametophyte. There is a discussion about that. And herbaceous, it had a creeping dichotomously branched rhizome. The rhizome was also dichotomously branched, bearing rhizoids on the underside. You can see the rhizoids, these fine hair-like structures. The aerial shoots arising from rhizome were also dichotomously branched, but it also produced adventitious branches. I talked about that. This is a dichotomy, and these are the adventitious branches. The plant attained a height of only about 20 centimeter, 18 to 20 centimeter. Yeah, one more thing. Small hemispherical lumps or disc-like structures were scattered over the surface near the base of the aerial stem, but there are no leaf-like structures present. You can see the stem is completely smooth. There is no innescence or outgrowth. These are small dot-like things that you are seeing. These are the hemispherical. This one can also be called hemispherical discs or lumps were visible. What were those? Those were, some people have considered that those were the archegonia or antheridia, and this was the gametophyte. In fact, this was the gametophyte of Rhynia major. This was the, some scientists have claimed that the Rhynia gonivoni was the gametophyte and Rhynia major was the sporophyte, because first land plants, their sporophyte and gametophyte, they looked very similar. Okay, so but that has not been accepted, as Rhynia major is not in fact the Rhynia. So let's proceed further. That there are hemispherical discs. What are those? We will talk about that a little later. Some of the aerial branches were terminated by sporangia. The plant also produced adventitious branches, the stele of which was not continuous with the main axis or stem. So they might serve as vegetative propagation on detachment. These branches, the adventitious branches. The characteristics of sporangia are as follows. The sporangia were oval or cylindrical structures with pointed ends present at the apices of the dichotomies. They're only 3 millimeter long. You can imagine 3 millimeter is how much? Just imagine how much will be a 3 meter, even smaller than a rice, small rice grain. They may be slightly constricted at the base. Here you can see there is a constriction at the base. Wall was thicker and multilayered with outer cells thick-walled, and no method of dehiscence could be observed. There was no, there was no slit anywhere on this, so it may be imagined that there was no predetermined line of dehiscence. It used to disintegrate at the time of dehiscence. The sporangia were filled up with spore tetrads or free spores. Okay, the structure of spore has not been shown here, but the spores must have been the trilate reticulate kind of exinus sculptures with trilate mark, trilate aperture. Anyway, this is the transverse section of this stem. I will show another diagram for this. Here is a simplified diagram from K.R. Sporne's book. There is a central xylem with central. This dot is protoxylem, position of the protoxylem. This dot-like structure is the position of the protoxylem, and these striations, that particular circle, it denotes the xylem strand. Outside that, this particular circle is the phloem. Outside that, this is the cortex. The cortex is quite wide. There is outer cortex, and there is the inner cortex. And there is in between them, there is a narrow zone of middle cortex, which is not always found but sometimes found, and this is a zone of darkly stained cells. So this demarcates between the outer and the inner cortex. The outer cortex is a little bit compact and the inner cortex is loosely present. The cells of the inner cortex are loose and they have larger intercellular spaces. Outside, this is the epidermis. Outside the epidermis, there was a cuticle layer, and epidermis was interrupted by stomata. So this diagram will show you that. So this is the cellular diagram of that. So you can see the stomata over here. So stomata are present in the stem. That means stem was performing the function of photosynthesis. Because there is no leaf, so of course the stem has to do that. And these are the inner layer of cortex, and they were, of course, had a larger air gaps between them, but this diagram does not show actually. I don't know. This diagram is taken from some book, and the Taylor's book possibly, but you should see the other diagrams, which you will find there are air gaps between them. And the outer layer is more compact than the inner layer. Why these gaps are there? Because this stomatal chamber, there has to transfer gases. Gaseous exchange has to take place. This particular inner cortex was photosynthetic. It contained chlorophyll, which is not possible to be preserved in the fossil. Chlorophyll or chloroplast will not be preserved in the fossil, so it is difficult to say. It can only be imagined because the air gaps were more in the inner cortex and they were connected with the stomata. So the function of photosynthesis was mainly done by the inner cortex, cells of the inner cortex. Okay, and the stele is very, very small, narrow. There is a very narrow strand of xylem surrounded by phloem. There is no visible endodermis or pericycle. It cannot be distinguished. It was almost merged with the inner cortex. So these are the anatomical characteristic of Rhynia. I'm not repeating this. Now, controversy regarding the gametophyte of Rhynia. This is a question sometimes asked as, you can say, the end questions, sometimes 8+2 type of questions, or 6+4 kind of, in the 4 marks tail end question is sometimes like this. What is the gametophyte? If the structure of this sporophyte is like this, what was the gametophyte like? So people talk, no gametophyte has been found for Rhynia. It is difficult to find a gametophyte. You can imagine, gametophytes are very softer tissues made of parenchyma cells, and the fossils, the process of fossilization is a very long time process. The plant will generally, the parenchymatous tissue will disintegrate, it will degrade, it will decompose very soon, so it will not be preserved. Very rarely some parenchymatous tissue might be preserved. Sometimes some cortical cells are preserved. Otherwise, they are also disintegrated, and scientists have to imagine that this empty area was possibly made of parenchymatous cells, and it may be cortex or pith or whatever. So these areas, the parenchymatous tissue will not be, gametophyte is made of parenchymatous tissue, so that those will not be preserved generally. So no, that's why no. But Marker, Pant, Lemoigne during this period, 1960s to 70s, have considered that the rhizome could have acted as the gametophyte. One of the ideas is this. Another idea, Lemoigne, what he said, that Rhynia gonivoni to be the gametophyte of Rhynia major. But as we now know that Rhynia major is not at all the Rhyniopsida anymore, rather it is considered a non-vascular plant called Aglaophyton major. So this question does not arise, that the Rhynia gonivoni is not the gametophyte of Rhynia major. Lemoigne described that the small lumps on the stem surface were, some people are quarreling outside. I don't know what is up. Anyway, let me concentrate. Lemoyne described that the small lumps on the stem surface were protruding antheridia or archegonia. If those were antheridia and archegonia grow on, these are sex organs, they grow on the gametophyte. So the small lumps, those were visible. If they were antheridia and archegonia, then definitely the Rhynia major, Rhynia goniboni, the sporophyte that we described, it is not actually sporophyte, it should be gametophyte. But those were not antheridia or archegonia. In support of his view, actually, he said that, Lemoyne said that the lumps, the hemispherical lumps that were visible on the basal area, these things, these dot on the stems, these were hemispherical lumps like structures, little bit risen over the stem, and he thought that those may be archegonia or antheridia. So if they were antheridia archegonia, so this was a gametophyte then. But later on it was found that those were actually secretory glands. They were revealed to be secretory glands. They were not antheridia and archegonia. So that was that particular structure was actually sporophyte. In spite of all these studies, gametophyte of Rhynia is still not described or discovered. So this is the discussion about Rhynia gametophyte. Okay, now let's talk about the Zosterophyllopsida. Zosterophyllopsida. Next group. Okay. Characteristic features of the class. It is also dichotomously branched, but sometimes pseudomonopodially. So dichotomous branching is primitive. And then pseudomonopodial means one of the branches overtopping the other. So it is becoming pseudomonopodial. And if that becomes the feature permanently, that always there is a branch which is overtopping the other, then from pseudomonopodial it will become monopodial, it should be called. Okay, the pseudomonopodial branching system. Sometimes H type or K type of branching evident. I will show you what is K type or H type of branching. Sporophyte is rootless and leafless. This is also rootless and leafless plant. Sporangia globose or reniform. They are not spindle shaped as was there in case of Rhynia. Lateral in position in spike. So this is not terminal in position, lateral in position, and in spike-like clusters. Spike is a kind of inflorescence. Spike inflorescence. In spike inflorescence, what happens? There is a stalk on which the flowers are attached, the sessile flowers without any pedicel. They are attached directly, laterally on the stalk. Okay, so that kind of thing. The spike-like clusters. Sporangia dehisce transversely along the distal tip, somewhat like Cooksonia. In cross-section, the xylem strand of aerial axis is massive. It is massive, bigger than the Rhynia. Elliptical to terete. Terete means circular. Elliptical means oval. And it was protostele. Xylem was exarch. In Rhynia, we saw that the xylem was centrarch. Here it was exarch. The genera included in this group was Zosterophyllum, Rebutia, Goslingia, Bathursia, Crenalicotis, Acollis. There are so many. So we will have to study only the Zosterophyllum. Now, talking about Zosterophyllum. Zosterophyllum is represented by many species, but none of them are worldwide in distribution. Zosterophyllum rhenanum was found from Germany, Zosterophyllum fertile from Belgium, Zosterophyllum australianum from Australia, Zosterophyllum minor from Russia, and Zosterophyllum myretonianum from Russia and Scotland. So Zosterophyllum, there are many species. As I said that the most important among them is the Zosterophyllum myretonianum. This has been studied most. Then Zosterophyllum rhenanum has also been studied quite a bit. Zosterophyllum fertile, this has been used to understand the fertile spike of the genus. So this is the structure. This is the overall physiognomy of the plant. So K-type or H-type branching, they were talking about that. This is H-type. See, you see the rhizome part. It is going. Some part is one branch is going aerial, another branch is going down. So you can see that H-type of structure, H letter is being formed, isn't it? Every time the branching is taking place, it is very much tufted. I mean, the branching are profuse, and sometimes it is K-like or it is H-like. Can you understand? This is K, looks like a K letter of English. This one looks like an H letter of English, isn't it? So that is what is called H-type or K-type branching pattern. Tufted growth. The aerial axis was dichotomously branched, sometimes monopodial, means pseudomonopodial. One of the branch is longer than the other. And as they said that the sporangia were oval to kidney shaped or rounded, and they were laterally attached. They were not terminal, and they formed a spike-like structure. This is what is that spike-like structure is this. Okay, if you and this particular diagram is of Zosterophyllum myretonianum, and this fossil Zosterophyllum rhynianum, it revealed that the lower part of the plant was actually submerged underwater because of the reason that the lower portion, the outline of the lower portion is little bit flattened. The upper portion is rounded, the lower portion is flattened, and the stomata are only present on the upper portion, they are lacking on the lower portion. This indicates that the part where from the stomata are absent and they are flattened, that portion was submerged under water or muddy area, I mean, and these spike-like structures, they were above the water level. And this is this diagram is of Zosterophyllum fertile. Here you can see the actual structure of this sporangia somewhat. You can put a question mark like structure. This is a question mark, this is a question mark, like this. If you draw it, drawing it is a little bit difficult. Just put question marks one above the other, it will become a spike-like structure. Can you see the question marks? Like this, little bit bent here and then this. Again a question mark, and like this you put question marks, okay, one after the other. And here there will be a little bit of, I mean, gaps are there, which if you can't do that, doesn't matter. While drawing, you can draw like this, what I'm saying. You need to draw during your description of the plant. So this is overall structure of the plant. We have to describe with the help of three different species, myretonianum, rhynianum, and Zosterophyllum fertile. Now, this is the description of the plant. The plant body can be differentiated into lower rhizomatous portion and upper aerial portion. In Zosterophyllum myretonianum, the rhizomatous portion showed H-shaped tufted branching system. Presence of stomata in the upper portion of the aerial stem suggests that the lower rhizomatous part of the plant might have been submerged. A reconstruction of the Zosterophyllum rhynianum by Krausel and Wieland 1985 further strengthened this evidence that the plant bodies was mostly submerged except the reproductive shoot, which was raised above the surface of the water, isn't it? As far as Zosterophyllum myretonianum is concerned, the erect branches were about 15 centimeter. We have to also say this, that how tall the plants were, 15 centimeters, smaller than the Rhynia gonivoni, or approximately similar, because you know, fossil records are rare. So by finding some fossil records, one cannot say 15 centimeter, 20 centimeter, not much different. I mean, some plants might be longer than this or shorter than this also. Tall and 2 millimeter in diameter. The central vascular strand is about 0.25 millimeter in diameter. Okay, how much it is difficult to imagine. but they are saying that the central strand was larger than the Rhynia. The aerial parts also showed lateral branching to some extent. They were there. Dichotomous branching was evident in Zosterophyllum renenum rather. The plant body is terete, means cylindrical, in vegetative axis and elliptical in fertile axis. Anatomically, the stellar organization is protostelic. The stele was also protostele with annular and scalariform tracheids in the xylem. Epidermis is cuticularized and bears stomata only in the upper aerial branches. This line, the plant body is terete in vegetative axis and elliptical in the fertile axis. I told a little earlier that the elliptical or flattened on the lower side and terete on the upper side. But here this point has been written opposite. So we will verify this later on, what is wrong with this. Because usually the submerged submerged plants are little bit flattened. From that concept, I said that. The lateral sporangia in the Zosterophyllum were grouped together in a terminal spike. This is the reproductive structure is spike-like, over which the sporangia are attached laterally. On the basis of their arrangement, Croft and Lang, 1942, have divided Zosterophyllum into two subgenera. One is Zosterophyllum and another is Platyzosterophyllum. What is the meaning? The former contains those species whose sporangia are spirally arranged. I mean, on the spike, on the main axis of the spike, the sporangia are present all along in a spiral manner. While in the later, named platy means plate-like, so sporangia are arranged in two rows, one or two rows, in two rows rather, mainly. Zosterophyllum myretonianum, renenum, australianum thus belongs to Zosterophyllum, whereas Zosterophyllum fertile, Zosterophyllum minor, and another species, Lanoveranum, etc. This possibly has, this was not there in the Sporne's book. This has been discovered later on. Belong to the Platyzosterophyllum. The spike of Zosterophyllum fertile has been well characterized. In this case, sporangia were present in two rows. Sporangia were ovoid to reniform and dehisced by means of slit running across the distal face of the sporangium. So this is the description of the plant. Here is the phylogeny of Zosterophyllopsida. Sometimes you are asked, what is the relationship with Rhyniopsida? So if you talk about that, both Rhyniopsida and Zosterophyllopsida, both are herbaceous with dichotomous to pseudomonopodial branching, both are protostelic, both have sporangia that are terminal on branches. This is not true exactly. Some of them may be terminal, but in case of Zosterophyllum, they are lateral in position. Here, in lateral position of the sporangia, it distinguishes it from Rhyniopsida. Both are homosporous, however. For above similarities, it is agreed that Zosterophyllopsida shared common ancestry with Rhyniopsida, but problem arises when the position of the sporangia are considered. Banks uses this feature as one of the important distinguishing features between Zosterophils and Rhiniophytes. However, the shape of the sporangia of Rhinophytes like Cooksonia and Renalia are globose to reniform, so which is quite similar to the Zosterophils. And transverse dehiscence give strong support to their relationship with Zosterophils. I mean, the Cooksonia and Renalia, we have not talked about Renalia. Cooksonia, at least I showed you the diagram. The structure of Cooksonia sporangia is similar to the Zosterophils. However, the position of the sporangium is always terminal in all Rhiniophytes. That is the difference. In Zosterophils, the position is lateral, whereas in Rhiniophytes, the position of the sporangia is terminal. In Rhynia, the sporangia is terminal position elongated with longitudinal dehiscence rather. The similarity between the lateral position of sporangia in the form of spike with the strobulus of the lycopsids suggests that Zosterophyllum represented a lateral plexus giving rise to the lycopsids with Cholangiophylum being at intermediate position. Now this is about evolution, that if you see the strobulus of Lycopsida, I mean Lycopodium or Selaginella, have you read that? Can you recall Lycopodium and Selaginella strobulus? They are very much like similar to this one. I mean, very much similar to this one, except that there are, I mean, they have a leaf-like structure below that, which is called sporophyll. I mean, if I can draw it, is there any pen-like structure over here available? Pointer, yeah, there's a pen. Well, so in lycopodium, you will see what? There is a axis, okay? Each of these, there is a kind of sporophyll-like structure like this. Then there is sporangia. Then again there is a sporophyll. Then there is sporangium. Then like this. This is the strobilus. On the other side also, here also there will be a sporangium like this. Then it will go like this. So this is a longitudinal section again. I'm trying to show you. So is there any similarity between these two? Of course, both are lateral in position. The only difference is this: there is a, I mean, the leaf-like structure below the sporangia in lycopodium. You know, lycopodia and selaginella, they are much evolved plant. Leaf has already come, but they might have come from Zosterophyllum kind of plant, Zosterophyllopsida group of plants. It seems from this particular structure that this similarity, it says that their plexus is same. That is what is being said here. So if Rhyniopsida is the primitive the first group of land plants, from there through Rhynialis, Zosterophyllalis, they evolved, and from Zosterophyllalis, Lycopsida, the group of plants, the Lycopsids plants evolved through Calamites. Another branch, the Sphenopsids, they came from Rhyniopsida through Trimerophytopsida. So now we will talk about Trimerophytopsida. Are you getting interest or not? You are understanding my teaching?
Yes, sir.
So,
Yes, sir.
okay, Trimerophytopsida, this particular, here again I have given is a class status to this, and in Sporne's classification it was an order status, Rhyniophytalis. Okay, let us consider this Rhyniophytalis. Okay, the Trimerophytalis, Rhyniophytalis, Trimerophytalis, Zosterophyllalis. This was the status, order status, according to K.R. Sporne, but other scientists, they have lifted them to the class status. So that's why it has been named like this, Trimerophytopsida. So characteristic feature, vascular plants, again, with the main axis that branched pseudo-monopodial. Now dichotomous branching is gradually being left. Gradually, because dichotomy is the primitive type of branching. From dichotomy, pseudo-monopodial, then monopodial, then sympodial. This kind of branching will evolve. The pseudomonopodial branching is gradually becoming more predominant and had laterals that in turn branched repeatedly, either dichotomously or trichotomous. The lateral branches showed dichotomy or trichotomy. Terminal paired fusiform sporangia present in some of the ultimate branches. Note this, paired and fusiform. Fusiform is more or less spindle-shaped. It can be called similar to the Rhynia, the shape of the Rhynia sporangia, more or less similar, but here they are present in pair and terminal in position, and we will see that they were suspended. I mean, they were hanging like structure. Okay, pendulent. Dehiscence of sporangia was longitudinal. They were also homosporous and protostelic. Okay, as far as this is concerned, they are similar. I mean, Rhyniopsida, Zosterophyllopsida, and Trimeropsida all are similar as far as the homospory protostelic nature is concerned. So there are three members of this: Trimerophyton, Psilophyton, and Pertica. We will study Psilophyton. There are many species of Psilophyton, like Psilophyton princeps, Psilophyton depsile, Forbacei, Psilophyton dawsonii, crenulatum, and Psilophyton microspinosum. So here that has been described is the Psilophyton princeps. Was the first species of this genus to be described by Dawson, 1859. 1859. See, before Rhynia was described in 1917 by Kidston and Lang, and here Dawson has described this plant much before, 50 years before that, from Lower Devonian, even earlier than this plant was there, and earlier than Rhynia. This will be rocks of the Gapse Peninsula, Quebec. This is the location from where actually this fossil was found. It is characterized by, now let us see the characteristic of the plant. This is how the plant looks like. If we characterize by the diagram, you can see that there is a horizontal rhizome on which you can see rhizoids are there. And the aerial axis, this is not exactly dichotomous, but some dichotomy and some pseudomonopodial branching, both are there. Okay, and there are several inescence, this spine-like or thorn-like structure that you are seeing. These are basically inescence, I mean outgrowths, and they did not have any venation within them. So they were neither microphyll nor they were spine or thorn, they were actually inescence. Okay, and these dichotomies, the lateral dichotomies, they were coiled like this, succinately coiled, particularly the sterile one. And as we said earlier, that there were sporangia in pairs, and they were fusiform and suspended like this. I mean the pendulant kind of. Okay. Now let's see the size of the plant, how big it was. The plant was slender, upright, dichotomous, stem somewhat similar to that of Rhynia, arising from the creeping rhizome. The rhizome was creeping. From that, the aerial axis arose. But the plant was 1 meter tall, very much tall, about 5 times taller than Rhynia. Okay, with the main axis being 1 centimeter in diameter. 1 centimeter, you can imagine more or less about the size of a tulsi plant, just for the sake of, imagination. Tips of the branches were terminally coiled. Some branches terminate with a pair of pendulant sporangia. On the lower parts, short, about 0.3 to 2 millimeter long, spine-like projections were observed. Now, what was there? It has been written in another point here. The axis is clothed with abundant inescence. This is not the characteristic of all the species. This was there in Psilophyton princeps only, which have been described as leaves, spines, and thorns, but their tips appeared to be glandular. They lacked stomata and also lacked vascular supply. So actually, they were neither leaf, nor spines, nor thorns. They were inescence. You might have heard about what is inescence. Inescence is basically an outgrowth. So here what we are seeing, what we are witnessing is evolution of leaf, microphyll. The Rhynia, it did not have any inescence. It was the stem was smooth. Zosterophyllum, the stem was smooth. But in Psilophyton, we see that there are some spine-like structure without any vasculature. So this is possibly the first step how leaf-like structure has started its origin. According to inescent theory, if you believe in inescent theory by Bauer, 1935, you see the inescent theory. You may write in your copy that you will see inescent theory, maybe through Google or in your book, textbook. What is inescent theory? That is interesting. Our inescent theory of origin of microphylls. Again, the xylem strand was massive. Where is the cursor? Okay. The xylem strand was massive compared to Rhynialis and was made up of scalariform tracheids. So these are the features of Psilophyton. Another, sporangia were borne in pairs. They were spindle-shaped or ovoid, terminal, and suspended on the ultimate dichotomies, relatively larger than Rhynia, 4 to 6 millimeter long and 1 to 1.5 millimeter thick. In case of Rhynia, it was only 2 to 3 millimeter long. The xylem strand was massive compared to Rhynialis and was made up of, this is repeated again. Stomata were present in the cuticle of the aerial axis. Therefore, it is presumed that the cortex was the principal site of photosynthesis. Here also, the photosynthesis, in fact, there is no leaf, so photosynthesis must be carried by this stem. So this is how the structure, this is inacen. This spine-like structure that you are seeing, this is called inacen. It has no vascular trace going inside. So this is the first step of origin of leaf according to inacen theory. This is the terminal sporangia, how it looked like. There is a dichotomy. At the tip of that, there are two fusiform sporangia suspended like that. This is scalariform tracheids that has been shown here, a part of that. So up to that, we have studied the Psilophytopsida groups. Psilopsida group, not Psilophytopsida. Psilopsida, the three members of Psilopsida, Rhynia, Zosterophyllum, and Psilophyton, already completed. Okay, is there any question from your side till now?
No, sir.
You are getting the, I mean, whatever being taught till now? You are able to understand?
Yes, sir.
Okay, so should I continue?
Yes, sir.
Okay. Lepidodendron, and there are two genera left now, Lepidodendron and Calamites. I will finish it within 20 minutes. I can see. I will try. Lepidodendron. This is now the Lepidodendron belongs to Lycopsida. If I go to this particular classification, Lepidodendron, it belongs to Lepidodendralis, this particular Lepidodendralis. Okay, and some classification actually keep it under, okay, this is under Lycopsida, of course, Lycopsids. Lepidodendron is actually Lycopsid. Selaginella is Lycopsid. Izoetis is Lycopsid. Selaginellalis is order. The genus is Selaginella. Another genus Izoetis. Another genus Lycopodium. These are there in your syllabus. All these they belong to Lycopsida. And Lepidodendron was a tree. It was a tree of this group, Lycopsida. There used to be trees under pteridophytes. Currently, the living pteridophyte, only two genera are present as tree form. If you try to find a tree pteridophyte, you will only see that there are two members, the Cyathus and Dixonia. These are the two tree forms. Other than that, there is no tree in pteridophytes. But earlier, these plants, the earlier plants, they were pteridophytic plants. They used to be tree-like in habit. So this was the plant. What is the characteristic? Lepidodendralis include the arborescent Lycopsid. Remember this word, arborescent Lycopsid. Because question sometimes, as I said, that the genus name is not given in the question. You might be asked to describe the arborescent Lycopsid given in your syllabus. Sometimes the question is asked like that. They want to know whether you know that the meaning of the word arborescent and Lycopsid or not. There is only one arborescent Lycopsid known, and that is Lepidodendron. Okay, arborescent means woody, tree-like, and Lycopsid means it belonging to Lycopsida group. That were most conspicuous element of Carboniferous period. You can imagine the time period, Carboniferous. Those were Devonian. We were talking about Devonian plants in the just before, regarding Rhynia, Zosterophyllum. Those were Devonian plants, and this is now Carboniferous plant. So members of this group are responsible to a large extent for extensive quantities of plant material that resulted in the formation of Carboniferous coal seams. The coal that we see, the coal seams that were formed during Carboniferous, they largely belong to this Lepidodendralis group. Okay. Remember that. Members of the Lepidodendron are characterized by the presence of secondary xylem, quite a good amount of secondary xylem, extensive peridome. That means secondary growth used to take place. These are the results of secondary growth. Those plants, those were that we talked about, the Rhynia, Zosterophyllum, they did not show any secondary growth. Their stele was protostelic in nature. Here the stele will become siphonostelic, also protostelic, but they will produce secondary xylem. As well as the the peridome is extrastellar secondary growth. Cortex is 3-zoned. Stigmarian rootlets, we will see this is the Stigmarian rootlet. So basically these are not true roots. They are somewhat similar to rhizophore of Senaginella. They are also dichotomously branched. You can see dichotomy of this. Okay, this is called Stigmarian rootlets. Single functional megaspore per megasporangium. This is interesting thing. Single functional megaspore means seed-like characteristic. In seed, the definition of seed is this: the ripened ovule is called seed, but in paleobotanical terms, a seed is a megasporangium containing a single functional megaspore that should be integumented and indehiscent. There are 3-4 words. 3 words, keywords, important. Integumented, it should be into a seed. Seed can be defined as structure which is integumented. First word, containing a megasporangium, single functional megasporangium means what? You may recall that during the megasporangium formation, this is the integument. You just recall. I'm just trying to recollect your memory, that there is a mother cell which undergoes reduction division that forms 4 cells like this. Then 3 of them become degenerate. Are you able to recall this? And one becomes larger, and then the nucleus divides mitotically into 8 nuclei. 3 on this side, micropylar end, 3 on the other side, chalazal end, and 2 in the middle. This structure, can you recall this?
Yes, sir.
This is present in the unfertilized ovule. After fertilization, this is the egg. These are 2 synergids. These are antipodal cells. These are central nuclei, polar nuclei. So this has been formed by a single functional megaspore. This is the sporangial structure actually. The sporangia contains a single functional megaspore, and that is integumented. This is what a seed is about, isn't it? So this is the structure of a seed. Seed is defined by these terms. Okay, why I'm emphasizing this? Because a little later we will see that Lepidodendron, the fruit body of it is called lepidocarpon. So there is a question whether lepidocarpon is a true seed or a false seed. Discuss. That will come a little later. I will talk about that. So these are and another thing is this, that integumented single functional megasporangium, and third one is this indehiscent. This feature. That means the development of mega gametophyte. This is the mega gametophyte that is being formed. It is endosporic. Within the spore, this is taking place. Dehiscent means the spore will be released from here, it will fall on the ground, and their germination will take place. The thallus formation or the female gametophyte formation. That is dehiscent nature. In seed, that doesn't occur. In true seed, this will not occur. It will be the integument is for protecting it and it making it indehiscent. So these 3 characteristics: integumented, indehiscent, single functional megaspore within the megasporangium makes a ovule or seed. Either ovule or seed. In case of paleobotany, it is difficult to say that this ovule has been fertilized or not, because you are seeing a fossil. By seeing a fossil, you cannot say the ovule has been fertilized or not. So ovule and seed are, these two terms are interchangeable in paleobotany. So now, where is this text? Okay. So Lepidodendron is the most important and elaborately studied member of this order. Others are Diphorodendron, Syncytidendron, etc. Earlier, all these were considered to be Lepidodendron. In earlier literature, there was only one. Later on, they have seen that it should be divided into two other genera. Anyway, this is another structure very important. In fact, the Lepidodendron was first described by this leaf base only. Lepidodendron was first described by the leaf cushion, external morphology of the stem. Fossils of this plant are preserved in various forms like casts, impression, compression, and even permineralization, all different kinds of forms. Morpho-anatomical features are like this. The habit of the plant was tree with trunks attaining a height of 30 to 35 meters. Imagine the height of the plant, 30 to 35 meters. Quite a tall plant. Okay. At the base, the diameter was 1 meter. The stem was armored by leaf cushions that were arranged spirally. The leaf cushions were characteristics of this plant. Why? We will see that. They were rhomboidal with vertically longer than wide. See, this is a single leaf cushion, and there were many such present side by side in a spiral way. Okay. So what are the structure? It is longer than wider. There is a vascular supply in the middle. Here you can see this one, VB. It is written VB. Sorry. VB, this particular structure, this one, and these two dots are flanking the vascular bundle. They are pericnose canals. Okay, pericnose scars, and there is a canal inside the base, and it used to go inside the leaf also. And these are also pericnose scars or pericnose canals. These are called infrafoliar pericnose, and this is foliar pericnose. Okay, here from actually the leaf was actually the petiole was attached, and above that this is the ligule pit. So these plants were ligulate plants. Ligule is a structure present at the axil of a leaf. It is not a bud or stem. It may be called. There is a discussion about ligule. So what is a ligule? A ligule, if it is a leaf, there cannot be a leaf at the axil of a leaf morphologically, but it is a kind of an outgrowth, you can say. So this is ligule pit. Here used to be a small, if the leaf was like this, say from here the leaf is growing like this. So this is the leaf of this plant. There used to be an outgrowth over here that was attached here. Understand? This is called ligule. You will find ligule in Selaginella. As a living member, in Selaginella, in the strobilus of the Selaginella, you will see that there is a ligule-like structure like this, and then the sporangium is present here. This is sporangium, and there is an outgrowth like this is this ligule. Ligule has a base like this structure, and this is the leaf like, the sporophyll. So this is the structure of the cushion of Lepidodendron. So what are pericnose? Pericnose were longitudinal channels traversing the length of the leaf parallel to the vein and are believed to be one type of aerating organs. Aerating organs mean gaseous translocation, gaseous movement used to take place through pericnose. Maybe they were also aquatic sometime, because similar to Arenchyma, Arenchyma is usually present in aquatic plants. There is a relation with these were trees. but there might be some evolutionary relationship with the aquatic plants. So those have been kept as a vestigial organ in these trees also. So this is the transverse section of Lepidodendron. Here I am describing by this diagram only, not reading this particular literature. I will give you this, I will supply you this, the literature part. Okay, so there is a protostele in the center. Okay, these dots are the protoxylem, and the middle part is metaxylem. So it is exarch in position. Surrounded by this part is possibly secondary xylem, or maybe also primary xylem. This is the secondary xylem actually, the wood, secondary wood we call. Secondary xylem is actually called wood. And this particular area was empty, possibly it was phloem. It has not been preserved. Phloem is relatively thinner cell wall, having thinner cell wall, so they disintegrate. This is inner cortex, this is middle cortex, and this is outer cortex. Okay, they are quite massive. The inner and the middle cortex, they contain leaf traces also. The leaves originate from the stem. Their traces belong to this inner and outer cortex. They're in the cross section, you can see that. Outer cortex did not have any leaf trace. Middle and the inner cortex. And then outside the outer cortex, there was a thick bark-like structure which is called peridome. Peridome is the extracellular growth that produces the bark of a stem. Okay, bark in fact is all the structure outside the phloem is bark. But peridome is only the outer layer of the bark, you can say. And these are the leaf bases, the cushion-like structure that I showed you just a little ago. So that part. So this is the cross section of the stem of Lepidodendron. So this is the structure of Lepidocarpon, the seed of Lepidodendron. Whether it was seed or not, that is the question. So there are two things. Why Lepidocarpon is considered seed-like, and why Lepidocarpon is not a true seed. So discussion are regarding both of them. Okay, actually it was not a true seed, but it looks like a seed. This is the fossil in which you can see this structure was thought to be integument. This is the integumented structure. It looks like that. There is a vascular trace here. This is the female gametophyte. This is the female gametophyte. Okay, surrounded by another covering. What was that covering? You can consider that this is the megasporangium, within which the megaspore was there, which has developed endoscopically, forming the female gametophyte inside. Then there will be archegonium archegonium formation on the tip, because this is a gymnosperm. I mean close to gymnosperm. Lepidodendron is pteridophyte, of course, but if it is a seed habit-like structure, it will form here archegonium. Within the archegonium, egg will be present, because in gymnosperm similar kind of structures are there. Those are seed plants. So if it is closer to that, scientists say that this is not integument. This structure is not integument. It is basically the sporophyll. It is leaf-like, foliar in nature. So it is not integument. This is not micropyle, because pollen grains have never been found trapped to this. This is not micropyle. And rather, sometimes it has been seen that the megaspore used to it was dehiscent. It used to come out of this and germinate outside on the soil. So if those things are there, this is not a true seed. As I said, there are three things should be there in the seed. It is a single functional megaspore. Okay, that is true. This part is okay. Even if you consider it is integument, it is okay. If it is integumented, you can consider it. It seems like that. So what is the discussion given here? Lepidocarpon is considered as seed-like. Why? Retention of megaspore. Retention of megaspore within the megaspore. The megaspore which develops into the female gametophyte is retained within the megasporangium. Here they are saying that it is retained, But in contradiction, when they will consider it as not true seed, they will say it is not retained. A dehiscent megasporangium. Opposite to retention of megaspore, the opposite point is dehiscent megasporangium. I mean, the megaspore was not retained, it was dehisced. So this is not matching with the thing is contradictory now. Protective enclosure, that is integument. The megasporangium is enveloped by a structure resembling an integument with a micropyle-like opening similar to the protective seed coat in true seeds. But nature of the integument was what? The integument-like structure is in Lepidocarpon is believed to be modified leaf, foliar in nature, not homologous to the true integument. So in this sense also, this is not a true seed in that way. Endosporic development, here the endosporic, the fertilization mechanism. Fertilization in Lepidodendron, they are not talking about endosporic development anymore. Some different point is there. Endosporic development was there. I mean, endosporic means what? The gametophyte was formed inside the spore wall. It is not it doesn't mean that it dehisced and then fall on the ground. That is not exosporic. If it falls on the ground also, endosporic development is possible because the gametophyte is being formed inside the wall of the spore. If that is there, that is endosporic development. Okay. But in situ or ex situ, that is the question. They are saying that it was dehiscent. That means sometimes it used to be ex situ also. The fertilization mechanism in Lepidodendron likely required water. This is the question. If it requires water with motile sperms reaching the archegonia, similar to ferns and mosses, so in that sense it is not a seed. In contrast, seed plants utilize pollen tubes to deliver non-motile sperm directly to the egg, eliminating the dependency on water. So on one side, when seed is being, we are defining a seed. In case of seed, the method of fertilization is through pollen tube. Okay. So, but if the method of fertilization is like pteridophyte or mosses, this cannot be as advanced as seed, cannot be said to be seed. Do you understand? So you have to write this discussion. If you're asked this question, you have to write this discussion. Now, with Calamites, I will finish this lecture. So Calamites is another tree-like plant. It is fossil. It belongs to Sphenopsida, not Lycopsida, Sphenopsida, the group in which the equisetum belongs, the living member equisetum. Okay, Calamites belong to that. Only one genus has been taken from there in your syllabus. And so Calamites were giant tree-sized horsetails belonging to extinct family Calamitaceae, thriving in Carboniferous to Permian. I mean, these were also from Carboniferous period, and they ran into the Permian also, swamp forests. How much old? 360 million to 250 million years old. I mean, in crores. 36 crores to 25 crores years ago, these plants used to survive when dinosaurs have not come. They could reach 20 to 30 meter tall, little smaller than Lepidodendron or more or less similar. Sporting segmented, but they were segmented like segment wise, they were divided. From nodes, leaves and branches used to arise in whorl like this. The generic name Calamites applies to the pith casts of stems. They were initially found as casts, pith casts, and branches. Stems and branches, the pith casts of stems and branches was first found, and they were given the name Calamites. The pith casts exhibit ridges and furrows corresponding in numbers to protoxylem strands. What is this? Ridges and furrows means, suppose this is the cast, this piece of cast. Okay, so on the cast you see that there are some, I mean, rhizine area, some are valley-like area. And this is the node. In the node, they used to be alternate. I mean, the furrow becomes the ridge and ridge becomes the furrow. Again, in the another area, the furrow becomes the ridge. So this way, the stem was, and if you see the cross section, these were like this, wavy. I mean, this is what is ridges and furrows are. Okay. So this was the structure of the stem from outside. The trunk used to be hollow at the internode and pitched at the nodes. The internal diameter was about 30 centimeter. The diameter is less than the Lepidodendron. Several subgenera of Calamites have been identified based on the size, position, and regularity of branches. The regularity of branches and their size, their numbers, they differed. According to that, there are subgenera. For example, in subgenus Calamites calamitina, numerous branches are produced at some nodes. In subgenus Calamites stylocalamites, produced fewer but larger branches. Similarly, a more regular branching pattern is seen in Calamites diplocalamites, where only 2 or 3 branches are produced per nodes. The most profusely branched subgenus is, however, the Calamites crucicalamites. So these are the subgenera, and they were named because on the basis of the differences of their branching pattern mainly. So this was the TS that I showed you last day in the practical class. Calamites, this structure was being shown, and this has been, magnified diagram of that. Only 2 of these vascular strands, they have been shown here. This is the protoxylem canal which has been disintegrated. This is called carinal canal. This is the characteristic feature of the anatomy of the Calamites plant. That carinal canals are there, but vallicular canals, you compare it with the equisetum. In equisetum, you will find carinal canals as well as vallicular canals. Vallicular canals are present in the cortex. They are present in the cortex. Carinal canals are present in the vascular bundle. They are actually protoxylem which has become disintegrated. Okay, and they are, a hollow structure, a canal-like structure they have formed. This is the secondary xylem, secondary wood, and this area is for primary xylem. Okay, in the primary xylem, the protoxylem is disintegrated, forming the carinal canal. This is the structure, and the pith is, there is no pith. The pith is hollow. And whatever you see is the secondary wood. Outside that, cortex must have been present, but those are not preserved. They are parenchymatous, so they disintegrate usually. So this is the TS. The TS of this stem show very little primary xylem, and most of the thickening was provided by the secondary xylem. This is the secondary xylem, most of this. And they are separated in different strands by medullary rays, also called wood rays, into separate fragments or segments of strands. Okay, the protoxylem was represented by carinal canals, and a small amount of metaxylem present was entirely centrifugal. The protoxylem canals occur only in the aerial axis and developed by the rupture of the first formed protoxylem tracheids. There will be D here, over here. So they are formed by the rupturing or disintegration of the protoxylem. The wood rays varied according to species, dividing the secondary wood into several segments. These are the segments. This is one segment. This is another segment. This is another segment. You can see there are many such segments here in this whole diagram. Entire TS of the axis. The tracheidal elements were with scalariform pitting or with circular bordered pits on the radial walls. So this is the characteristic of anatomical feature of the Calamites stem. So that's all about these 5 different fossil member of the pteridophytes included in your syllabus. Thank you so much.
Sir, I have a question.
Okay, ask your question.
Sir, in Lepidodendron, what is the function of pericnous spheres?
Pericnose, I told you, pericnose canals, they are basically aerating channels. They used to run parallel to the veins of the leaf as well as the inside the stem. Okay, in the leaf cushion, they are visible as a scar. but actually they were canals or channels, you can say, inside the petiole and then the lamina of the leaf, and also in the stem, where it goes to the leaf trace, goes and attaches with the main stele, I mean the vascular bundle. Up to that, those channels were there. Okay, so those were actually aerating organs. What was their function? An aerating organ is usually present for gaseous exchange, but here it seems that they are some kind of vestigial organ, because this plant is not an aquatic plant, it is a tree, but this particular feature is an aquatic feature. It is similar to Arenchyma. Do you remember Arenchyma? Arenchyma is usually present in aquatic plants. Those are, the intercellular spaces that becomes larger. Sometimes they are formed by the disintegration of the cells, and they're also channel-like structures, usually present in the aquatic plants for, gaseous circulation, etc. So that is what is the function of pericnose. You got it?
Thank you, sir.
Any other question? I think you have understood it clearly. If you have any question, I will post this. I will give you the entire PowerPoint, or maybe the theoretical part of it in a separate Word file with the diagram as a literature, which you can consult later on. And I will take a small exam on it also in MCQ type questions. Okay, that you will get later. So now I'm stopping the class. Is it okay?
Yes, sir.
Okay, good night. How many of you are there? 24 now. Many have left. Chalo. Okay, I have taken a screenshot also as an attendance. And later on, when we meet in the class, we will take attendance for this in the register. Okay.